Hi, everybody. I'm Andre Bayet. I'm the Executive Director of MOHCCN. I think you can recognize many people from the team. Isabel, who's our Managing Director, and then people in communications. I'm not going to name everybody. And also various program managers, Jesse and company. But of course, also Véronique, who's going to be co-chairing with Natalie Quadrants, who's a very precious member of our council, very helpful, very smart. Very useful at these meetings. Thank you, Natalie. And today is the second of this reborn series of seminars. So I'm sorry I missed the last one. I had the flu. I was in bed. I had the real flu. And now I feel better. And so today it's the turn of MOHQ to present us update on their research. I will let Natalie and Véronique co-chair. They're the co-chairs of this event, so I will let them take this from here and move ahead.
Merci beaucoup, André. Bonjour, puis bienvenue à tous. Merci de nous avoir rejoints aujourd'hui pour, comme André a dit, notre deuxième séminaire de 2024, présenté par le consortium de Marathon de l'Espoir au Québec. First of all, I would like to wish everyone a very happy DNA Day. This seems like an auspicious day to be having a cancer genomics seminar. So I know we're all really excited to hear some great talks. I do want to take a moment just to acknowledge that I personally am calling you today from Mi'kma'ki, the traditional and ancestral territory of the Mi'kmaq people. Our network members and our speakers and our attendees today are located all across the country on lands that are home to diverse First Nations, Métis, and Inuit populations. And I invite everyone to think about what that means in their region and the importance of those people and those lands. I would also like to pick up on André's comments and introduce my co-chair, Natalie. As André has mentioned, she's been a member of the Network Council as well as the Network's Patient Working Group for the past year. She has generously volunteered to participate in our event today on top of all the fantastic work that she and the whole Patient Working Group network, Patient Working Group are already doing for the network. So we're very, very grateful to have you here with us today, Natalie. I will pass it to you if you'd like to tell us a little bit about yourself.
Thank you. Bonjour, hello. Je m'appelle Nathalie Andrés Quadrants. My name's Natalie Andrés Quadrants. Although I currently live in Calgary, based on my accent, you may gather that I came from Quebec at some point. I was born and raised and completed my Bachelor of Science at Concordia University in Montreal in the mid-90s. And then I moved out west to join Team Canada's snowboard racing team between 1995 and 1998. Fast forward a few years, in 2019, I was diagnosed with de novo stage 4 triple positive breast cancer. Et donc en 2019, j'ai été diagnostiquée avec un cancer du sein triple positif, de novo stade 4. My cancer has been inactive for the past 2 years, except it recently reared its ugly head again, and I just received my final pathology reports yesterday evening, confirming that it is indeed cancer again. Donc, malheureusement, mon cancer est redevenu actif après un congé de 2 ans, ce qui a été confirmé hier avec mes résultats de pathologie. So before officially kicking off the agenda with the consortium overview, I'd quickly like to introduce our 2 speakers on this. We have Dr. Anne-Marie Messe-Masson. She is a professor in the Department of Medicine at Université de Montréal. She's the scientific director of basic and translational research at the Centre de recherche du Centre hospitalier de l'Université de Montréal. Lots of centers in that one. Then we have Dr. Murag Park, a distinguished James McGill professor in the Departments of Oncology, Biochemistry, and Medicine, the Diane and Salguirera Chair in Cancer Genetics, and the director of the Rosalind and Morris Goodman Cancer Institute, all of which are at McGill University. Both Drs. Messe-Masson and Park are consortium leads for the Marathon of Hope's Quebec region. They will jointly give us an overview today of what's happening within the Quebec consortium. So welcome, doctors, and I'll pass it over to you.
So thanks very much for the introduction. And I think somebody has to share slides now, and I believe that's Louisa. And first of all, I want to thank Louisa, Christine, and Veronique, who are our program managers. Veronique, who's handed over to Christine. And what I want to say profoundly is without them, we would not be where we are today. As we can see, Marathon of Hope Quebec is a consortium. Could you give me the next slide? And it was a consortium at the beginning. So this came with all the challenges of building a consortium, and I'm sure the other consortia that have now formed can highlight and showcase, what that challenge is. What's very important from our perspective is we brought together 4 hospitals in Montreal, to CHUM and Hôpital Maisonneuve-Rosemont at the University of Montreal, the Jewish General Hospital in the MUHC within the McGill umbrella, together with our Cancer Institute, the Rosalind and Maurice Goodman Cancer Institute in the Genome Center. This had its challenges, but also its rewards. And what's important to highlight here is between these 4 hospitals, they see together nearly 22,000 new cancer patients every year. So this really brings a rich cancer ecosystem to the table and in the Montreal environment. Could I have the next one? Our philosophy in building our consortium, and this started with our pilot project, was to integrate multiple technologies, recognizing that the genomic landscape, of course, is very important to highlight the changes that occur in cancers, but that's not sufficient. And so within our pilot project, we highlighted the importance of immune profiling. This progressed into using massive cytoph imaging mass cytometry, where we can look at multiple immune markers, but as well as tumor antigen profiling to highlight how these tumors will respond to therapy and how they respond to, importantly, the new checkpoint type therapies. In addition to this, we recognize the importance of the metabolic landscape, the microbiota landscape, new technologies in microfluidics, for example. But really, one of the biggest strengths in our Quebec consortium was the long-term biobanks that we had established, and Anne-Marie will highlight some of the strengths of these over the last 10, 15 years that were supported by the Réseau Cancer in Quebec, and our live biobanking with patient-derived xenografts. So we've integrated all of these concepts into our consortium. Can I have the next slide? Our pilot project, as I highlighted, started October 2018. This was spearheaded by Ian Watson and John Stagg, 2 young investigators. And they together brought a project to the table focused on melanoma, but integrating the immune landscape and immunoprofiling. So this really kick-started us in this more multidisciplinary type approach. And it's important to highlight that immune profiling is now being included in many centers as a key component together with the genomic landscape. So what you can see here, we started October 2018. By April 2021, with Princess Margaret and BC Cancer, we achieved our designation as a node in the Marathon of Hope. By August 2021, we were granted our ethics approval for genomics to obtain the genomic sequencing of patients. Now, what's important here is we have a pan ethic approval for all the sites that are within the Marathon of Hope Quebec node. We submitted our first case March 2022. Important to highlight that we did not have a pipeline already established in the Marathon of Hope Quebec for genomic sequencing and the return of data. So this was a milestone for us. And I think a bigger milestone was by December 2022, we had already submitted our first 500 cases. By 2023, October, we were granted approval for sharing data, ethics approval for sharing data within Quebec. Some of you may know there's new legislation coming out in Quebec that will require additional discussion to share data outside of Quebec, but we now have ethics approval to share data within Quebec. We submitted our 1,500 cases by March 2024, and we're very proud of that, and that took a lot of work. And we now have our first Quebec node, the candid deposition structure set up. Anne-Marie will tell you a little bit more about the structure of that and how data gets entered into this and is seamless with the patient data. Can I have the next one? So just to give you a little highlight on the numbers, as I just said, we've submitted 1,500 cases since March 2022. We've got over 70 principal investigators. These are a mixture of researchers, clinicians, and scientists as well, where members of our network participate and lead 30 cohorts across the Marathon of Hope network. And within the labs and within the clinical landscape, we've got over 170 highly qualified individuals, project managers, bioinformaticians. And the importance is coordinating this network across multiple sites. And these individuals are indispensable. Could I have the next one? What we're very proud about is also we've been successful in the applications for both the Clinician Scientist Awards and the Health Informatics and Data Science Awards. We've had 5 young clinician scientists are awarded Marathon of Hope Clinician Scientist Awards. Importantly, to highlight is that they reside in all of the different hospitals that are part of our network. So that includes the CHUM with Bertrand Routy and Huda, Philippe Lefrançois at the Lady Davis, the Jewish General Hospital, Natasha's at Hôpital Maisonneuve-Rosemont, and Basil, who's our latest member, is at the MUHC. So each of the sites across the node are well represented. And we have 2 individuals funded in the health informatics, Parsa and Emmanuel, who are both at the Jewish General. Could I have the next one, please? What I want to highlight here is that these are the different areas that we lead, different members lead into the Marathon of Hope. You can see that we're involved with multiple different cancer types. Importantly, what I want to highlight is you can see by the filled in dots, that for each of these cancer types, for the most part, multiple sites are involved with these cohorts. So if you look at breast, for example, which I'm involved with, that's GCI in yellow, the MUHC, the JGH, and the CHUM are all part of that cohort. And this, I think, is one of the strengths of building a consortium and bringing together multiple sites. Our other strength was, of course, as I highlighted, that we had a rich biobank history in the Quebec landscape, and we've been able to use both retrospective and prospective cases where we have the full clinical information, and that was very important from our perspective. We're interested to look both, obviously, at the genomic landscape, but to really begin to study mechanisms of progression, particularly mechanisms of resistance. And we have a focus on rare cancer subtypes. I think the next slide, I hand over to Anne-Marie.
Thank you, Morag. So in addition to the clinical, to the genomic data, a really important aspect of the program is the clinical annotation. So in the Quebec consortium, we identified ADAM as the tool to be able to collect it. So those who don't know, it's a software suited for biobanking, where you have the ability to include studies, It is centered around the participant who provides the sample. We have the clinical annotations, and as well as all the samples and collections we get. And for this particular project, then, we have created an MOH version of the ATOM where the dictionaries for the clinical data match those that have been defined by the MOH study. Next, please. So how does it work in Quebec for us to be able to deposit and share data? So within each of our sites, we have an ATOM MOHQ install. In the sites, in many of the sites, and we're bringing more on, there are data lakes where we can actually go and get that data from the institutional data lake and then populate the fields for clinical data. At the same time, on the research side, we populate the sample registration of the specimen, so we know what specimen is going for sequencing. If the clinical data is not in the data lake, then we can enter it manually, or at sites where they have not yet connected the data lake to the MOHQ ATOM, ATOM users then can enter the data as per the instructions. That data then is through this install sent to the CanDig, Montreal CanDig site, where it can bring together the sequencing data, and the visualization is done through Bento. And in addition, at that point, some of the data is returned to the ATOM MOHQ so that we can follow what happened to samples in terms of quality control and moving forward through the sequencing. Next. So this is just an overview of the clinical data annotation, so we can fill in the fields for any participant in terms of diagnostics, treatments, and follow-ups. Next. As well as for the data, as for the return of information, for example, the MOHQ sequencing manifest, then we know when the sample was sent. We know which flow cells it was run on. We know all the QC information and went forward, and one of the major results in terms of quality of the sample. Next. And in fact, once the data is now ingested into the CanDig, then there is through Bento a visualization tool, so you can do federated searches if you want to understand what the different cohorts are, what kind of data is present in the Quebec node, as well as clicking to individual participants. Next. So all of this couldn't have been accomplished by just a few people. The teams are quite large, but we did want to highlight, first of all, we have a steering committee that meets monthly to help shepherd this project through. As Morag pointed out, we have very talented and hardworking project managers. And at each of the sites, we have site coordinators too to help us ensure that the samples are reach the Genome Center in a timely fashion and the clinical data is there to join. Of course, the Genome Center itself as the sequencing lab and a bioinformatics team that support this project. We have an ATOM MOH team that ensures that the sites have access and are busy creating the connections between the data lakes that are present and ATOM. There's a data wrangling team that has been working of how to flow the data then from the ATOM MOH into CanDig. And of course, there's numerous staff involved in the collection, review, and preparation of samples and all the associated clinical data. And we can't forget that behind that, there are a whole research administration teams that have been working at contracts, at ethics, and finances to ensure that each of the MOHQ sites. And finally, our partners, not the least of which is TFRI, Health Canada, our patient partners who are an important, and our funding partners, because as you know, this is a co-funded initiative. So we have many funding partners that have helped us reach this goal. So with that quick overview of the MOHQ, and the next slide, I'd like to introduce our two speakers. So Dr. Elkrief, who recently joined the SHUM, as she mentioned, is going to be presenting on the role of the gut and intratumoral microbiota on the response to immune checkpoint inhibitors in lung cancer and melanoma. And we have Juan Miguel Romero, who is from George Zogopoulos' lab, who is using unsupervised clustering of multiomics data to define molecular and transcriptional subtypes of rare pancreatic neuroendocrine tumors. So I leave it up to our presenters to continue.
Great,
Hi,
thank you.
I'm just going to quickly jump in before our presenters continue. I wanted to introduce, but you have done a fantastic job, so I'm not going to repeat everything you've said. But I wanted to kind of kick it off for Dr. Elkrief. When I read your abstract, I was really fascinated by the concept of using fecal transplantation to treat lung cancers and melanoma. To me, that just blew my mind. It kind of reminded me of a story when I was a business strategist and I was working as a partner with Microsoft to launch their new Vista platform. And right up until the launch, it actually had a code name called Project Longhorn. And the way that name came about was there were a few executives who were skiing at, doing some après ski actually at Whistler, the base of Whistler, and there was a bar at the bottom of the hill called Longhorn. And that's where they paper napkined their concept. And so that was the code name they used until launch. So I'm really, really looking forward to kind of understanding how this study came about, as I find it's a really interesting and not naturally intuitive as a patient concept. So really looking forward to hearing from you, and I'll pass over the mic to you.
Great, thank you so much. So I'll be sure to highlight a bit of the story aspect as I talk about it. And so to conclude, we're really excited about these microbiome clinical trials. We're looking at ways to shape the gut and tumor microbiome. And we're really excited about how to incorporate next generation sequencing with Terry Fox in these research projects to be able to look at the tumor microbiome in many different ways. And this is a lay summary of the project. And I'd like to acknowledge collaborators, especially members of the Rudy Lab, for whom the clinical trials would not be possible, and members of my lab, which is a brand new lab that just started in December, and all of our funding sources, and of course the Marathon of Hope. And last but not least, my mentorship committee, Anne-Marie Mesmasson, Dr. Fabre, and Ian Watson, and Bernard Lemieux. Thank you very much.
Thank you so much. That's like incredibly fascinating. With that, I'm going to let Véronique introduce our next speaker. Thank you so much for your update.
Thank you, Natalie. And yes, I'll echo that. That was a great presentation. Thank you, Dr. Elkrief. Next up, we have Juan Miguel Romero. He is an MD PhD candidate in the Division of Experimental Medicine at the Rosalind and Morris Goodman Cancer Institute and the Research Institute of the McGill University Health Center. Today, he will be telling us about his work in George Zogopoulos' lab, as Dr. Mesmasson mentioned earlier, looking at molecular characterization of rare pancreatic neuroendocrine tumors. Juan Miguel, please take it away.
Thank you. Let me share my screen here. Okay, so first of all, thank you to the MOH for the opportunity to present our work. So hi, everyone. I'm John Miguel Romero, a fifth-year MD PhD student in the lab of Dr. George Zogopoulos, and I'm here to present on an early analysis of the Quebec Regional Consortium pancreatic biliary malignancies. Originally, the talk I had submitted was focused on maybe a more scientific or a more nuanced approach. I decided to maybe take a step back and give a more clinical overview, and the second half of my talk will focus on that specific aspect. So the main subtype of pancreatic biliary cancers is pancreatic cancer, and the main subtype of pancreatic cancer is pancreatic ductal adenocarcinoma. It's the most lethal solid malignancy in Canada with the lowest 5-year overall survival at 10%. It's projected to become the second most common cancer-related death, and a systemic disease early on, with the majority of patients diagnosed at late stage. There have been few advances in treatment options with little increases in cure rates. Therefore, new treatment strategies are urgently needed. As we can see here in yellow, preventability and incidence are moderate, And this is mainly because risk factors like obesity and smoking can be controlled, and the low incidence because it's rare, but it still has poor survival, detectability, and a high mortality. So we've learned a lot about the pancreas cancer genome, particularly through collaborative efforts across Canada. Mainly what we've learned is the importance of biobanking and data sharing, which is very important, particularly for this rare type of cancer. But we still need to understand these cancers better. So this talk focuses on discussing how this MOH Quebec pancreatic biliary cohort has focused on addressing this via two aims. And following Dr. Elkrief's excellent talk on a similar avenue, how we can use this data retrospectively to better analyze these tumors and understand the immune landscape, to better stratify patients that can receive immunotherapy. And this ties in back with my PhD, which has focused on a signature of tumor immunity and ICI response, which we'll discuss near the end. So it's important to provide point-of-care tests to help identify potential genetic mutations that can help inform patient treatment in this cancer. And how do we do this? Through the Quebec Pancreas Cancer Study, we sequence or we do panel testing on all incident cases of pancreas cancer where they receive germline testing. And we've leveraged the MOH platform to take these patients and then comprehensively analyze their full genomes and transcriptomes to better characterize their disease. And we are applying these findings where possible to help inform patients in the clinic to better guide treatment strategies. Bit of a background on pancreas predisposition. So pancreatic cancer is primarily sporadic with unknown genetic drivers. The highest predisposition group is familial pancreas cancer, which are families that have high incidences of pancreatic cancer. And the most prominent syndrome for this is hereditary breast and ovarian cancer, which is mainly due to BRCA2 and BRCA1 mutations. And indeed, what we find is that germline mutations, which means that the mutations can be passed on from offspring, from parents to the offspring, is that in BRCA1 and BRCA2, these two genes are the most common genetic causes of pancreatic cancer. And this is important because BRCA1 and BRCA2 are involved in this process of homologous recombination. Briefly, when cells are unable to repair double-strand breaks, we can then exploit this clinically by giving drugs that can target this pathway, and specifically two drugs, platinum and PARP inhibitors, are used to treat these patients that have these BRCA1 and BRCA2 mutations. And this is data from the seminal POLO trial, which showed that the efficacy of a PARP inhibitor called olaparib did show use in BRCA mutated pancreatic cancer. But what's interesting is that 17% of patients had progressed on cisplatin, which was a type of platinum therapy, which had to have been given before enrollment in this study. So together, this suggests that even though germline mutations can exist, their cancers are not BRCA driven, nor are they dependent on BRCA mutations. Through a collaborative effort, we also showed that while germline mutations can lead to this HRD phenotype, about 12% of these germline carriers lacked the second mutation needed to demonstrate HRD. Therefore, what does this mean? It means that germline testing is a good surrogate for HRD, but it's not perfect. And we need a tumor biomarker to confirm HRD specifically, not just look at germline BRCA1 and BRCA2 mutations. The only robust tool available currently is genomic sequencing. And then this brings us back to the MOH on how we can use this data for a clinical case to better inform patient treatment. So this case is a 7-year-old male that presented to the clinic with lethargy, hyperglycemia, and weight loss. Through point-of-care germline testing via the Quebec Pancreas Cancer Study, we identified that they had a pathogenic germline BRCA1 mutation. We did CT imaging and liver MRI, and we found a 2-centimeter resectable mass in the body of the pancreas. The fine needle biopsy was found to be a moderately differentiated adenocarcinoma. And this patient was subsequently enrolled in the CCTG PAC3 trial, which is a phase 3 trial studying neoadjuvant chemotherapy versus upfront surgery. And they were randomized to receive upfront surgery. And here's a CT scan. They had a distal pancreatectomy, and here's a graphic schematically on the right. Essentially, if there's a tumor at the tail of the pancreas, we can resect that portion of the pancreas to then leave behind intact pancreas to avoid morbidity changes. And what we found was that when we reviewed the pathology, it was indeed early stage pancreas cancer, moderately differentiated, negative margins, and 3 of the 29 lymph nodes were involved. And this patient went on to receive full course adjuvant FOLFIRINOX for 12 cycles. But unfortunately, a year later, they progressed, with painful supraclavicular lymph node. And here's just a diagram. It's not the actual patient. It's just for illustrative purposes. And then this patient received FDG PETs. And what's important here is you could see these two nodules glow up, and we weren't sure whether this was a new primary or metastatic disease. So this patient had biopsies done on these two, and essentially this was confirmed to be metastatic primary from the primary pancreas cancer. So based on this, how do we treat the patient? We have 4 options. We can re-challenge on platinum, and if we see a response, we can give maintenance PARP, which is what we do for tumors based on the POLO trial. We can just give a PARP inhibitor. We could give the standard of care of gemcitabine and nab-paclitaxel for PDAC, or do we use the MOH data? And of course, this being a talk on the MOH data, I think the answer is clear. We use this data to try to understand whether they do have hallmarks of HRD. So using this report, we looked at the whole genome sequencing and we confirmed that they did have a BRCA1 mutation that was found on our panel testing. But interestingly, when we used an algorithm that can better identify HRD, we found that this patient indeed did not have HRD. We need a score greater than 0.7. And on top of this, another surrogate, which we can determine from whole genome sequencing, it was also absent. So response to a platinum and a PARP inhibitor was unlikely. And so based on this, there was no evidence of HRD, and the patient was therefore treated with standard of care chemo, avoiding unnecessary treatment. And they ended up receiving tailored therapy based on their tumor profile. So this is an example of how the MOH was used prospectively to help guide patient management. Provides real-time clinical information, and it helps decide optimal chemotherapy regimens, particularly for these BRCA2-driven pancreas cancer. Now, as I said, my particular interest and focus of my PhD is in understanding T-cell inflammation in different subtypes of pancreas cancer. And many recent papers have shown that HRD and BRCA mutations may make tumors more immunogenic, and current chemos may in fact work in an immune-mediated way, particularly these PARP inhibitors, which I discussed. And recent data further suggests that HRD may harbor an immune hot phenotype in pancreas cancer specifically. So we wanted to find a good marker of T-cell inflammation in pancreatic cancer. And we found that a 4-chemokine signature consisting of CCL4, 5, CXCL9, and 10 was associated with a higher expression of genes and gene sets involved in the cancer immunity cycle. And we need the cancer immune cycle to be activated for immunotherapies to work. We further validated this signature across cancers using 4 independent data sets, with tumors displaying varying expression of these scores, and we found that it was able to predict T-cell inflammation. Furthermore, it could predict response to immune checkpoint treatment in different cancers, which is something that we could not show in the previous study for lack of immune checkpoint data for pancreas cancer. Not only that, it did a good job at predicting this when compared to other published immune signatures. This paper here was particularly exciting because it showed that when you combine, it combined pancreatic biliary, HRD, NRC score, and it found that patients that had response to immune therapy had increased expression of these 4 chemokines. So this score may have broad applicability not just in pancreas cancer, but in other cancers and HRD cancers as well. And now this brings us to the second part of our talk, which is identifying patients that could respond to immune checkpoint inhibition by understanding their tumors prospectively, particularly as it applies to PNETs. Why PNETs? PNETs are rare tumors. They have a variable clinical behavior and prognosis. They have durable responses that are often difficult to achieve, and the subsets of PNETs may have microenvironments that make them amenable to immune checkpoint inhibition. So we want to know, how does the C-score fare in PNETs, and how does this compare to pancreatic ductal adenocarcinoma, the main subtype? So there's emerging data that shows a role for immune checkpoint inhibition in PNETs. Here, these top papers show that when they looked at an immune checkpoint inhibitor called pembrolizumab in PNETs, they had some responses. And interestingly, one patient that had an ongoing response of over 24 months was a PNET. And then the other 2 papers show similar data as well, particularly this one that showed a prolonged response again in a pancreatic neuroendocrine tumor with immune checkpoint inhibition. In addition, groups have looked at the genomes of these tumors, and they've found that approximately 17% of patients have germline mutations, including in genes like BRCA2. So this all ties back to our clinical interests of BRCA2 mutated pancreatic biliary cancers. So what's our cohort? We looked at a cohort of 34 patients currently as part of the MOH dataset, with a mean age of 53 ranging from 24 to 82 years old, approximately half are female. And we've received the data from 22 whole genomes and 15 whole transcriptomes, which is what we'll show subsequently. And here's a brief cartoon highlighting again the differences between neuroendocrine tumors and exocrine tumors, where the former arises from the endocrine cells and the latter arises from the exocrine cells. And here we're focusing on the endocrine part. So using this MOH platform and the data, the rich data that we get, we can obtain multi-omic data, meaning multiple genomic, transcriptomic, et cetera, avenues to better interrogate different aspects. So here we're looking at the somatic or the tumor mutational profile. This is an oncoplot that gives a high-level summary of the somatic profile of these tumors, where each column is a patient and every row is a gene. And here you can see the fraction of patients that have mutations in said genes. And then each color represents a specific type of mutation. So for example, blue represents missense mutations, et cetera. And here I want to highlight two specific genes. The first here is the MEN1 gene, which in our data set, we found to be mutated in 52% of the samples. And in the previous report, they've shown that 37% of the tumors had this mutation. So it's in line with these findings. And we may in fact have selected a population that has a higher proportion of mutations in these tumors, which we can further explore. And second is this mutation in a gene called PRSS1, where over 40 mutations in this gene, cause hereditary pancreatitis. And this is known to be linked for pancreatic cancer. It increases the risk, but interestingly, it's not been found to increase the risk for pancreatic neuroendocrine tumors, which is interesting and we'll delve deeper. So we can also use this MOH platform to look at whole transcriptome sequencing, and we can use clustering to try to differentiate tumors based on their profile. Here, kind of avoiding fancy jargon, we can use an algorithm called unsupervised clustering to identify two different groups of patients, and then we can use differential gene expression to show the differences in genes that are expressed in these two groups. Here's a heat map showing what these differences in genes are. And again, rows are genes and columns are patients. And you can see from the annotations, this was cluster number 1, this was cluster number 2, and you can see differences in gene expression. This column shows the genes that are involved in some way to be processes of the immune system. And here you can see that quite a few genes have roles in the immune system. And then here, only focusing on these genes, you can see again that there are clear differences between groups 1 and groups 2. But you'll also notice that we have an annotation here for the C-score. And we can see that these 4 patients that clustered as immune hot also clustered on this group that was also identified by this unsupervised method. So now let's take a step back and let's just focus on these C-score high versus the C-score lows. And we can compare the expression of these immune-related gene sets. And what we could see is that patients that had high expression of these signature had increased expression of gene sets involved in the cancer immunity cycle, specifically CYT or cytolytic activity, which is a marker of T cell activation and a few other cells, interferon gamma activity, which is a process linked to T cell function, and then the gene set involved with T cell inflammation. However, lower numbers preclude more robust changes, and we're collecting more data to help differentiate these two groups. And then finally, how do PNETs compare to PDACs? So here we added a subset from the PanCure dataset of 106 resectable pancreatic ductal adenocarcinomas. And here we're looking at the expression of the chemokine score with PNETs on the left and PDACs on the right, and patients marked as either high in red or low and medium in blue. And here we can see that the PNETs seem to have an intermediate expression of the C-score compared to the PDACs. With PDACs having both lower and higher expression of these patients. And interestingly, in a paper that recently profiled the tumor immune environment of PNETs, it seems that PNETs may seem to be slightly lower on the immunogenicity scale on PDACs, and we seem to be finding similar results. So to summarize this patient, we've used unsupervised clustering to identify two groups based on RNA-seq data. These clusters have differences in immune gene expression. And we've also divided patients based on their C-score, and we've shown that these patients have higher immune-related signatures, and we have deeper interrogation of somatic and germline mutations underway. We're continuing to collect these PNETs, and by the end of the year, we estimate hopefully to have about 50 pancreatic neuroendocrine tumors, which would be a good data set to look at further. And then finally, the summary of the presentation here is that we're using the MOH platform on our regional cohorts to better understand these hard-to-treat hepatopancreaticobiliary cancers, specifically to profile patients with germline mutations for potential actionability in the clinic, to study the incidence of germline mutations and somatic landscape of PNETs, and if they drive the cancer, and then finally, explore tumor immunity, specifically the C-score in these pancreatic malignancies. So here are our acknowledgements. I'd like to thank everyone involved, particularly my supervisor, George, our lab members, as well as collaborators and funding sources. Thank you.
Thank you very much. That was a wonderful talk. So we will open it up to questions now. For anyone who has questions, please either write them in the chat or raise your hand and we can call on you. And I think Natalie has one to get us started. You're muted, Natalie.
There we go. I do that all the time. The first one is for Dr. Elkrief. What have patient reactions been, or how has the recruitment process been? Has it been difficult given that, at least from a patient point of view, it is an unusual line of treatment? And it's kind of a twofold question. With that, would you have any advice to other oncologists who are trying to enlist their patients into the studies to help them get a positive response from their patients?
So, in these clinical trials, so across 6 centers, we've enrolled now more than 65 patients. So we haven't had any difficulty with accrual from the patient perspective. I think that it's an experimental treatment, just like any other experimental treatment that needs, un consentement libre et éclairé in order to make sure that, the patient understands the risks associated with it. We do inform patients that the capsules are triple encapsulated, and so they're completely odorless and tasteless, and they're also designed for release in the small intestine, so there's no risk of it opening up earlier than it needs to. And generally the patients have been really excited about using a treatment that's been approved for another setting and trying to see if it can work in the context of cancer. So the feedback has been positive.
Wonderful, thank you.
Dr. Park?
Yeah, thanks very much. This is a question for the second speaker, Gwen. So there are multiple neuroendocrine subtypes of different cancers. Has anybody actually done profiling in a similar manner to what you've done? So you've looked at pancreatic neuroendocrine tumors. Has anybody looked at, say, breast neuroendocrine or others with respect to the immune status or other changes?
It's a good question, Dr. Park. So my focus really has been on the pancreatic portion. And really, even to complicate things further, pancreatic neuroendocrine tumors can be divided even more into insulinomas, lipomas, etc. So here, this is a more broad approach, just looking at the pancreatic portion of it. As you said, there are other types of neuroendocrine tumors, and I think a question is still whether we should group them all as generally neuroendocrine or whether we should also look at their histological subtype. A lot of these studies, at least the ones that I've looked at for immunotherapy, they're usually basket trials because these tumors are so rare. They collect multiple neuroendocrine tumors, and they end up looking at different types of histological cancers as well. So the collection of pancreas, gastro, etc., breast, I'm sure as well is included with varying responses. In terms of the genomic profile, I can't answer to that specifically. My knowledge isn't there yet. But for response to immunotherapy, it is variable. It seems like from at least 2 studies that I've seen, pancreas cancer does decently. And for another study, it seemed that thoracic neuroendocrine tumors did good on immunotherapy. So I'm sure that must also correlate with an immune landscape or a genomic landscape that people are still interrogating. And I think more work has to be done. And we're trying to fill that niche with the pancreatic portion.
Yeah, maybe that's something for us to think about from an MOH perspective with respect to rare tumors, if we can bucket neuroendocrine somehow prospectively going forward. Yeah. Thanks.
Thanks.
Audrey, I see you have your hand up. I did receive a question from another one of our patient working group members in the chat, if you're okay if I ask that one first. This one is back to Dr. Elkrief. The question is, they're wondering if there's a gut microbiome link with other cancer treatments, such as targeted therapies. Or is it just for immunotherapy?
Yeah, so we're looking at that now, particularly in the context of patients with melanoma that are treated with the RAF/MEK inhibitors, because we know that these targeted therapies also influence the tumor microenvironment. And so this is something that we don't have the answer to, but we are looking into it. And then in terms of other cancers, so gut microbiome modulation has been extensively studied in hematologic malignancies in the context of graft versus host disease, which is a complication of bone marrow transplantation, now CAR T therapy, and also kidney cancer. So it's being explored in multiple different kinds of cancers. It's a great question.
Thank you. Andre, over to you.
Yes, thank you very much. Both presentations were excellent, and of course also Morag and Anne-Marie Masmasson. That was also fantastic. Thank you very much. I have a question for Ariel, or two questions. The first one is sort of a chicken and egg kind of thing. So when you look at the tumors, you find E. coli, and then you tend to find more immune cells. Now, my recollection is we all have bacteremia, right? Regularly. I mean, we have circulating bacteria. So I'm wondering whether if there's any idea of which one came first. So are the bacteria there because there's some inflammation, and then the blood vessels are a little porous and they sort of home to the tumors, or the other way around? Is there any evidence either way?
So we're looking into that now. It's something that we're studying in the lab. I think there are two hypotheses. Is it a local colonization of just contiguity with the airways, and these bacteria come from the mouth, and fecal oral transmission, which we know happens all the time. Is it a direct translocation from the gut? Is it the macrophages that are bringing these bacteria to the tumor? These are all questions and hypotheses, and we're trying to model this in mice, and we're trying to test this out in the lab, and writing a grant right now to try to answer those questions. So yeah.
So I have another question. It's kind of silly. So we've been talking about fecal transplant for many, many years, right?
Yes.
It's usually from patients, right, or from normal donors or something like that. So
Yes.
I'm wondering why this cannot be done like not in a recombinant way, but with bacteria grown in a lab, and then instead of from feces, you just pull them and then you give them a pill, and maybe people like that better.
Yes. So ideally, yes, but unfortunately, all of the biotechs that have tested a strategy like that have failed. And so we don't know if it's just because selecting the strain. So recently there was a paper in Nature Medicine that for the first time looked at strains, because we know that within a specific species there are multiple different strains, and each strain has its own impact. So it could be that. It could also be the way that the bacteria interact with each other and the metabolites that they produce, because in the end, maybe it's the metabolites, maybe it has nothing to do with the bacteria. So we're working on that. We're working on trying to cultivate bacteria from the donors and try to identify these strains and then develop capsules. So yes, this is something that we're actively working on, especially with Bertrand's team.
All right, thank you very much.
Thank you. I see we're at the top of the hour, so we'll keep it to one more question. George, you have one?
Yes, thank you. It's a question for Ariel. I was just wondering if you had any correlation with antibiotic exposure prolonged. I mean, how are you going to rationalize that? Even things that everybody has a PPI now from some reflux, and it may affect your microbiome. Do you have any thoughts? How do we rationalize that? And
rationalize in terms of what do you mean by that?
Understanding the data, right?
Yeah.
So your microenvironment will probably have been influenced, similar to Andre's question. I just have a hard time grasping, how much of it is inherent and how much of it is something that we have done, and can we do something different in treating these patients beforehand or perioperatively and so on.
Yeah, so we haven't yet explored the relationship between antibiotics and the tumor microbiome. In terms of the gut microbiome, the relationship's pretty clear, but the tumor microbiome, we don't really have the answers to that question. So it's something we'll be exploring. We do know that the time window aspect is something that's very important. So in the 30 days prior, antibiotics are bad, but after, not so much. So it seems like that critical window is important. Whether we can safely delay starting immunotherapy, never been studied, so it's not something I recommend before it's studied, but you know, there's different things that we can imagine. There's a paper hopefully coming out of Bertrand's lab soon that shows the ability to sequester antibiotics with activated charcoal. So that's a potential strategy that's obviously still experimental, but something that's, you know, we're going to look into.
Hey, thank you so much, everyone, for all the great presentations and the interesting questions and the great discussions, and for joining us today. We really appreciate it. Our next seminar will be in a couple months, hosted by the VC Consortium, so we'll make sure to let all of you know when that's happening. Thank you very much and have a great day.
Thank you.
Thank you. Take care.